Question number 1 says, what are enzymes? The answer is C, biological catalysts. Now, question number 2 says, enzyme speeds up reaction by. The answer is by decreasing its activation energy. The region of the enzyme where the substrate binds is called. Question number 4 says, enzymes are specific to. The answer is all of the above: the temperature, the substrate, and the pH. Which of the following is not a property of enzyme? The answer is they change the nature of the product. Enzyme activity is affected by. And the answer is all of the above: the temperature, the pH, and the substrate concentration. Question number 7 says, an enzymes become denatured when. The answer is when its structure is altered. Question number 8 says, the substance upon which an enzyme acts is called. The answer is substrate. Question number 9 says, the enzyme substrate complex is formed when. The option is C, when the substrate binds to the enzyme. Question number 10 asks, which of the following is an example of an enzyme? The answer is B, amylase. The optimum temperature for most human enzymes is. The answer is B, 37 degrees. Question number 12 says, a competitive inhibitor. The answer is B, the one that competes with the substrate. A non-competitive inhibitor binds to. And the answer is a different part of the enzyme. Question number 14 says, enzymes are composed mainly of. The answer is C, proteins. Question number 15 says, the enzyme that breaks down starch into sugar is. The answer is amylase. Question number 16 says, which enzyme breaks down the protein? The enzyme that breaks down the protein is C, protease. Question number 17 says, coenzymes are. And the answer is, they are organic molecules that helps enzymes. The rate of an enzyme activity increases with temperature up to a point and then it decreases rapidly. Enzyme activity is measured by. The answer is substrate disappearance or product formation. Question number 20 says, which term refers to the loss of an enzyme's functional shape? And the answer is B, denaturation. Question number 21 says, which factor does not affect enzyme activity? And the answer is light intensity. Question number 22 says, enzymes work by. And the answer is C, by lowering the activation energy. The feedback inhibitions occurs when. The answer is B, when the product inhibits the enzyme. Question number 24 says, a cofactor is. And the answer is, a cofactor is a non-protein helper. Which of these is an example of a coenzyme? The example of a coenzyme is C, NAD. The enzyme lowers the activation energy by. By stabilizing transition states. Question number 27 asks, a zymogen is. The answer is B. It is an inactive enzyme precursor. The enzyme substrate specificity is often compared to. The answer is B, key and lock. Question number 29 says, the first enzyme discovered was. It was D, urease. Question number 30 says, the enzymes in the stomach works best at. And the answer is C, acidic pH.